Have a good number of attendees, so why don't we get started? My name is Jeff Lotz. I'm a professor at UC San Francisco in the Department of Orthopedic Surgery, and I'm the PI of one of the backpack mechanistic research centers who's hosting this seminar for today. I have a few Zoom housekeeping items before we get started. As Jake mentioned, if you wouldn't mind changing your name on the Zoom affiliation with your name and affiliation so that we can know who's participating and particularly who's asking questions. I'm going to be fielding questions at the end, so if you have questions, please enter those through chat, and I'll then organize those and pass those along to Laura. And Jake Williams and Daniel Davis are going to be co-hosts of the Zoom, so if you have any technical difficulties, you can do a private chat to them, and hopefully they can help you sort out any problems you might be having. So we're fortunate today to have Dr. Laura Stone. She's a professor at the Department of Anesthesiology at the University of Minnesota. She has training in neuroscience, biochemistry, and a focus in research on pain mechanisms, epigenetic regulation of chronic pain, and pharmacological and non-pharmacological methods of pain treatment. We've been fortunate within Backpack to learn a lot from Laura and her team, who are leading the Quebec low back pain study, which is a web-based cohort of acute and chronic back pain patients that's meant to collect baseline information and act as a referral network for satellite hypothesis-driven data collection centers. One of the tangible products of our collaboration so far is we're actually working contract with the same marketing group they've used to enroll and attract patients, and we'll be using them for our back home digital cohort. So thank you, Laura, for making that introduction. They have you taught them a lot that we're going to benefit from. And given our closely aligned objectives with Laura and her group, I expect that we'll have some close collaborations between her and the Backpack Consortium, sharing data and models as we go forward. So please welcome Laura today as she discusses her topic, which is epigenetics and chronic pain, what it is and why should I care? Thank you, Laura.
Thank you so much. Can you see my slide?
Yes.
Wonderful. Great. So I'd like to start by thanking Dr. Lutz and the Backpack Consortium for inviting me. I'm very excited to be here today to share with you some of our work on epigenetics and chronic pain. And I hope that by the end of the hour, if you have a bit of a sense for what epigenetics is, if you're not already familiar with it, and why it might be something interesting and important to study, then I will have definitely accomplished the goal for today. So as a note, my department of anesthesia is hiring a basic pain scientist and a pain clinician. So please check out our website if you're looking for something. We're very excited to expand our team here. So epigenetics, what you see here is the life cycle of a butterfly. And it takes on clearly very different phenotypes throughout its lifestyle, its life course. But the genome is the same. So as important as genetics and genomics are, there's other factors that influence the phenotype of an organism. And one of the main sets of mechanisms that dictate this change in phenotype is the control of which genes are expressed, which genes are turned on in a different tissue at a different development stage, what genes are turned on. And some of that is controlled by epigenetic mechanisms. Similarly, if you think about pain as a life course or a life cycle, we all have throughout our lifetime, we take on various phenotypes. And things that happen to us perhaps early in life might influence how we evolve later in life. And we carry some of that early life and previous experience with us. So what I'm going to do today is kind of walk through each of these steps, the role or the potential role of epigenetics in setting up risk and resilience factors for chronic pain. I'm going to provide an example of how epigenetics can drive an acute tissue injury or tissue pathology that could be pain generating. We're going to talk about the transition from acute to chronic pain and how living with chronic pain can alter the epigenome and how we might be able to target some of those pathological changes. We'll talk about treatments. I'll give an example of both a pharmacological and a non-pharmacological treatment for chronic pain. And then finally, in the last section, we'll talk about outcomes, and that's where I'll transition to primarily the ideas that we're working on for human studies in epigenetics. So epigenetics kind of literally means on or around genomics. So epi being on or around. And it's a set of mechanisms that don't change the genome, but can change the expression of genes. And in addition to developmentally programmed changes in epigenetics that might control how one cell becomes a heart cell and another cell becomes a neuron, we're also constantly being influenced by our previous experiences and our environment, what we eat, whether we're physically active or not, whether we've experienced previous injury or trauma in our lives. All of these things come together to set your current epigenome. I'm going to give you an example of why this is so important using a kind of classic example from the literature. And this is an example that demonstrates how an earlier experience can embed itself into our epigenome to dictate how we respond to things later in life. And in this example, this is work primarily done by Moishe Sif and Mike Meany at McGill University. They're the pioneers in this field. So if you take rat mothers, you can have either good rat mothers or bad rat mothers. And one of the defining characteristics of a rat mother is whether or not they spend a lot of time licking and grooming their pups versus not. So those of you who are already familiar with this, this body of work will know that pups who have low licking and grooming mothers, so they experience some early life, they have low maternal care or impaired maternal care when they're young. They tend to have increased stress responses as adults when they grow up and then are challenged with stressful situations. And they have decreased resiliency. Whereas pups who had good rat mothers will grow up to have a more adaptive, healthier stress responses. And over many years of really phenomenal work, it's been worked out that one of the things that drives this difference, this impact of maternal care on the behavior in the adult, is the expression of the glucocorticoid receptor in the hippocampus. So what you see here is glucocorticoid receptor expression in pups that had a good attentive mother, and some pups that did not have an attentive mother. And this receptor, the GR receptor, is really a critical part of the stress, the HPA feedback axis. So the implications of this are that if there's less glucocorticoid receptor expression, there's an altered feedback of the HPA axis, which leads in these pups to impaired resiliency. So this is meant as an example of how our early exposure can lead to a persistent behavioral impact once we're adults. And the mechanisms by this that drive this have been investigated extensively. And I'm going to talk about the role of epigenetics in using that as an example initially. So this is a cartoon of a gene. So it gets wrapped up on these little balls called histones and then packaged into chromosomes. And two of the major epigenetic mechanisms that can control or modulate gene expression are DNA methylation, and that is just what it sounds like, the addition of this methyl group to directly to, it's a CG residue So these CG residues either have methyl groups attached to them or not. And the other major mechanism are histone modifications, which are changes to these little balls that the DNA wraps around. And what happens is the DNA gets wrapped around them, and it's a pretty tight wrap. So in order for that gene to be transcribed, you need to kind of open the, you need to loosen the wrap around the gene that needs to be transcribed. So the tightness of the wrap can be controlled by these different histone modifications, including acetylation, phosphorylation, and methylation. Sometimes when people talk about epigenetic mechanisms, RNA interference, the role of non-coding RNAs is also included. Today, I won't be talking about that, and I'll primarily focus on DNA methylation. But just keep in mind that there's a growing literature on the importance of histone modifications in chronic diseases and treatment as well. So this slide is not as bad as it looks, and I promise will be the only one that looks anything like this. But I just want to point out a little bit more about how DNA methylation might work to change gene expression. So what we have here are some DNA wrapped around histones, what these little, I don't know what you would call those, like rolls. And in order for a gene to be transcribed, normally a transcription factor binds, and then the gene is transcribed. Well, in this example up here, these methyl groups have been added to the place on the gene where the transcription factor needs to bind. So the gene's not made because it can't get initiated. In another set of examples here, this methyl group, you can kind of see it hiding behind there, attracts a family of proteins called methylated DNA binding proteins, which then set in motion this whole pylon of really complicated molecular signaling, the net effect of which is normally to block gene expression. So while it's not always true that increased methylation means less gene expression, because this is biology, so of course nothing is always 100%, that's generally a useful way to think about it. So going back to the impact of early life adversity on the glucocorticoid receptor, the investigators in this case have taken this to human studies and asked the question whether they could reproduce the finding of the change in expression and the role of methylation and histone modification in humans. And so there's a brain bank at the Douglas Hospital at McGill University, and they have taken brains, in this case it's hippocampus, from suicide completers that were either abused as children or not. So this is the human equivalent of impaired maternal care. And they saw the same change in the glucocorticoid receptor. And they were able to demonstrate in adult humans that if they had been abused as children, there was also a change in methylation in the opposite direction that might be driving that altered resiliency in the HPA axis. So to take this back to pain, we have a situation here where early life or previous experience is impacting methylation in the brain, gene expression in the brain, and potentially behavior, at least in the animal models. And it's well known in the pain literature that there are many risk factors for chronic pain, including adverse childhood events, socioeconomic status, low socioeconomic status specifically, and other environmental impacts like chronic stress, depression, anxiety, and previous trauma, either pain-related or not pain-related. So it kind of suggests that there might be a role for epigenetics in embedding these risk and resilience factors that will play into how an individual responds to a pain insult later on. In order to study this in a preclinical model, to start to understand how whether we can directly link an adverse early life event to a change in pain, we did a set of experiments looking at the impact of life experience in rodents. And specifically, we went to very, very early life experience and looked at the role of prenatal stress on the subsequent pups after they become adults. So in this experiment, we received pregnant females. They were subject to a stress paradigm that is well established in the literature. They produce offspring. We let the offspring grow up so that they're fully adult for 3 months. And then we did a mouse model of nerve injury, which is called the chronic constriction injury. So this is an injury of the sciatic nerve. And then we tested their response to behavior to see how much that nerve injury produced pain. And then we collected in the brain regions and looked for changes in the machinery that sets DNA methylation. And what we found, first the behavior, so we have 4 groups here. We have individual mice who received a sham surgery so they're not nerve injured, but they were or were not stressed prenatally. And then there's the animals that were nerve injured. And at baseline, there's no difference. So the prenatal stress had no impact on whether the animal was just basal sensitivity to, in this case, it was mechanical stimuli. On the y-axis, we have their threshold, their sensitivity to mechanical sensitivity in the von Frey assay. On the x-axis, we have weeks. So what we found after injury, however, is that the animals that had nerve injury and stress, which is the solid triangles, were much more hypersensitive after the injury than the animals that had injury but were not stressed in utero. So this is an example of how a very early effect in life or event in life will lead to potentially increased sensitivity to the perception of pain or sensitivity to stimuli in the adult. So this is, by this point at the end of this experiment, it's multiple months after that original in utero stress. There's a really interesting, this is an aside, but there's a really interesting cohort study also based in the Douglas Hospital at McGill University called Project Ice Storm, where there was a really bad ice storm about 15 to 20 years ago now in Montreal. And everything was shut down for 2 weeks and you couldn't get anywhere. Like the whole city was covered in ice. And it was one of those life events that everyone who lived in Montreal at the time remembers. So there's a cohort of individuals who were in utero during the time of the ice storm, where their mothers would have been very stressed for about a 2-week period. And the groups of King and Sif and others have shown that those individuals who were in utero at the time are, who now are maybe 15 or 20 years old, have increased risk of obesity. They have some other impairments, and there is very noticeable differences in methylation in their blood. So there's actually a signature of that prenatal stress in these now adolescent offspring. So I hope that they continue to follow them because it'll be very interesting to see if they're susceptible to other chronic diseases as they age, like cardiovascular problems or even chronic pain. We asked the question about whether there might be a role for the epigenome in this phenomenon. And in this case, what we did was we extracted the hippocampus from the mice after the end of the experiment, and we measured the RNA expression of some of the enzymes that control the amount of methylation. And there's a couple interesting things that we found. The first was that it was much more likely for gene expression to be altered by the experience of prenatal stress rather than the 6 weeks of chronic pain. So here the solid bars are the prenatally stressed animals, and these are not injured and these are injured. And what you can see is that it's the effect of prenatal stress that causes this big increase in the amount of DNMT, which is one of those primary enzymes that adds methyl groups to DNA. We found other, you can refer to the paper if you want to see some of the other changes that we found, but the effect of prenatal stress was very profound in the hippocampus. We also found some examples where the combination of prenatal stress and nerve injury had an impact. So here, there's no difference in the control uninjured animals between stressed, not stressed, and stressed, but there is in the injured animals between not stressed and stressed. So this is just really meant as an example of the epigenome encoding a memory of this early life experience, and that embedded memory preset some of these animals to be more sensitive to the nerve injury than others. Up until now, I've only talked about individual genes. The thing that's really interesting about these big changes in the enzyme that adds methyl groups, for example, is that this could affect the expression of possibly hundreds to thousands of other genes, because it's one of those enzymes that controls expression of a lot of different things. So rather than just thinking about one gene like the GR receptor, it's really important when thinking about epigenetics to think about the whole genome and all of the things that might be changing together. So in this example, we find an experiment that was done in macaque monkeys that had either normal maternal rearing, which are these 4 here, or had a surrogate maternal rearing. So this is a model of, again, of maternal separation. And each of these rows, each row is a gene or a promoter, and each column is an individual animal. And the color indicates the degree of methylation in the different, in that promoter, in that animal. So what you can see by separating those who had surrogate rearing versus maternal rearing was very different patterns of methylation. So this is yet another example of that embedding of a memory, but now it is many genes, not just a single gene. And to take it further, this does not only happen in the central nervous system. In the same set of animals, there is also a pattern of over 100 different gene promoters in T cells that were differentially methylated, depending on the maternal rearing. So the implications here are that the epigenetic dysregulation could be persistent. It's also system-wide, and it raises the possibility of there being potentially peripheral biomarkers of previous life experience that might be useful as markers of risk for developing chronic pain, for example, or of sensitivity to different therapeutic interventions, for example. So that's kind of a very big claim, but it's really interesting to think about. So moving into acute pain, we've been working for a while with a mouse model of disc degeneration in which they're missing the gene called SPARC. And I probably should have said this at the beginning, but given this audience and the expertise in pain in general, and specifically in back pain, I have skipped all of the the normal introductory stuff about back pain affecting this many people and all of that. We all know it's a huge, huge problem. So this is essentially a model of accelerated aging, where these mice have disc degeneration and show signs of axial and radiating pain. And here we see, this is a measure of their disc height. So they have reduced disc height. But we also see this in just aging wild type mice. So this is, I think they were CD1 mice at different ages, and just part of the natural aging process, there is a decrease in disc height. There is also, in the spark mice, an increased sensitivity to cold stimuli, which is shown here to here. If you, we can talk about how this test works during the question period if there's time. And we see the same increase in aging wild type mice. So we asked the question, is it possible that in normal mice, as they age, the spark gene might be decreased, thus making them more susceptible to disc degeneration and pain? So here we have measured spark expression in the aging mice, and did indeed find that it was decreased as they aged and as their discs became more degenerated. And we looked at whether or not there was a change in methylation of the gene itself that might be driving these age-related changes. And what we found here is the short answer is yes. These numbers indicate how far a particular CG residue is from the transcription start site of the gene. So the gene expression will go this way. And there were multiple sites that are becoming increasingly methylated, resulting, we presume, although we haven't been able to conclusively link them, but consistent with, you might say, this decrease in the spark gene that is probably driving disc degeneration, at least to some extent. So that was really interesting, but we were curious, well, do you see this in humans? Because this is just a bunch of old mice. So we collected, in order to test that, we've worked with a number of collaborators to collect human discs from either patients with back pain that are undergoing surgery, or control discs from transplant donors. And the transplant donor initiative has been driven largely by Elizabeth Haglund from McGill, who many of you may know. What we found in the human gene was this very similar pattern of increased methylation in the painful degenerating discs compared to the control discs. So at the time that we published this, it was the first demonstration of epigenetic control of a single gene being linked to a human pain condition. There's still a lot more work to be done here, though. We've very much just scratched the surface of understanding the epigenetic components that drive disc degeneration. So that is an ongoing area of interest. One of the really exciting things about epigenetics, and as opposed potentially to genetics, is that the epigenome is malleable. It's possible to change it by changing experience, by changing environment. And there's quite a bit of literature on how lifestyle, like diet and exercise, can influence the epigenome. So we were wondering if we could change the status of the discs in the spark mice with therapeutic exercise. And in this experiment, we're measuring acetone sensitivity. So this, again, is cold sensitivity. You can see the spark mice here are more sensitive at baseline than the wild type mice. And we gave them home cage wheels, so they have the opportunity to voluntarily run whenever they want. We just left the wheels in their cages. And the animals that had functional wheels, it took 1 to 2 months, but the cold sensitivity completely reversed in the spark mice. So we've been asking questions lately about what has changed in these discs that as a result of therapeutic exercise? And are those changes things that maybe we could take advantage of therapeutically with either drugs or to target more specifically? And I'll just give you a couple examples. One of them is overall a change in the total amount of methylation in the discs. So what you see here on the y-axis is total methylation of all of the DNA that we took from the mouse discs in wild-type animals that were either sedentary or had exercise, or spark animals that were either sedentary or had exercise. And we've done this in both males and females. And the interesting thing is that exercise is decreasing global methylation. So we would expect, thinking somewhat simplistically, but we would expect that there would be an increase in overall gene expression in these discs. Perhaps the disc is trying to rebuild itself. And we've actually shown increases in several, like in aggregin and a couple of collagens as a result of this exercise. What I'm going to show you today is an example of the spark gene itself, where we measured how much spark expression there was, and fortunately there was no spark expression in the knockout, so that was reassuring. But several months of running did increase the spark gene therapeutically in wild-type animals. So that might be an opportunity for genetic therapy, or maybe just a really good argument for increased physical activity in individuals that have back pain associated with degeneration. Okay, so moving to chronic pain. So far we've talked about how early life experience can set your epigenome to maybe have an influence on how we respond later in life, and how epigenetics might drive some pain-generating pathologies. But living with chronic pain, the experience of chronic pain itself can also change the epigenome. So we've focused our studies trying to understand how pain influences the epigenome. We've focused it largely on the prefrontal cortex, based mostly on human brain imaging studies that have shown changes in the cortex in when you compare individuals with chronic pain to those without, and specifically the dorsolateral prefrontal cortex showing some signs of recovery of anatomical changes or pain-related changes in brain activity after therapeutic interventions. So the work that I'm going to show you for the next few minutes is all in the frontal cortex. So we suspect that epigenetics has a really important role in this neuroplasticity because it is reversible in some situations. And epigenetics is reversible. So this was our hypothesis, that there is an epigenetic memory of chronic pain. And in order to start to address that, the first experiment we did in mice was a model of nerve injury. In this case, it's called the spared nerve injury. It's a branch of the sciatic, or two branches of the sciatic are cut. We allowed the injury to mature for many months. And an important side is that I personally believe that it's really important in these animal models to allow the model to evolve for several months to more fully represent chronic pain in clinic. A week after an injury in a rodent is not equivalent to a year after an injury to a human. And what we found in the frontal cortex, our first sign that something might be going on, was a change in the global methylation in the cortex. So we wanted to know, well, okay, there might be a change in global methylation, but is individual genes changing, and how broad are those changes? So we did an experiment, in this case in rats, 9 months after nerve injury, again in the frontal cortex, where here you see another heat map like the one we saw earlier, where each column is an animal and each row is a gene that was differentially methylated. And when you take the methylation data from all of the animals and all the genes that were shown to be differentially methylated, and you throw them into a computer and you say, can you group these samples by common similarities? We get something like this, where all of the sham animals clustered together. So we didn't tell the computer who was who. They just clustered based on similarity. And all of the nerve injured animals clustered together, except maybe one guy who's kind of off to the side a little bit. But when we did this cluster analysis, there's this quote from our collaborator that just really, to this day, hits me. And he said, you know, this is relatively rare, this amount of clustering. They normally see it when they're comparing different tissues or species. So these rats had the same environment. They were genetically similar, if not identical. Same food, same life. The only difference was whether some lived with chronic pain or others. And there's a massive change in global methylation and patterns of methylation in their brains. Like in the macaque maternal care study, we also looked at T cells and found similar clustering in T cells. There weren't as many genes in T cells that came up, but they were also very strongly clustered. So living with chronic pain does not just change the brain, it's also changing the immune system and possibly many other systems in vivo. One of the really interesting things here was that we were able to find a set of 11 different methylation sites in the genome that were fairly predictive, taken on their own, as to whether or not an animal had pain or not. So here we had the nerve injured animals, and based on the 11 methylation sites, every animal got a score as to whether we thought it was going to be a nerve injured or not nerve injured. And if you were to look at like a cutoff of 0.5, it's right most of the time. So that was a really exciting proof of principle for us for the idea of epigenetic biomarkers in humans later on. So I'm a little bit worried about time, so I might skip a few things in the next section, but we're going to talk about treatment for a few minutes, first in the animal models. And one of the first experiments we did with pharmacological interventions as proof of concept for changing the epigenome as a therapy was using this over-the-counter nutritional supplement called SAMe. So SAMe is an endogenous compound that is the endogenous donor when a methyl group is transferred to DNA or proteins. So we all have SAMe in us all the time, but people take it as a supplement. I got this picture from the Costco website. It's marketed for mood, for osteoarthritis, and for cognition. So we modeled an experiment based on how a person might use this over-the-counter drug. So we started with adult mice that had already had nerve injury for several months. They got the drug orally 3 times a week for 8 weeks. So it was a long treatment period. And the nerve injury was already existing. So they'd already had chronic pain for several months. So this is very much a treatment paradigm. And the short story of what we found was that it did in fact improve their mechanical sensitivity, but it didn't really kick in until about 2 months into the treatment. So this isn't a magic pill that you would give someone and they would immediately have an effect. This is kind of a chronic, slow shifting, perhaps, in the epigenome that has a therapeutic benefit. I'm going to now skip a couple slides and get to this point where we looked at the brains of these animals that had had SAM. And we had a comparison between what were all the differentially methylated genes between injured and not injured, and that was this many. And what were all the differentially methylated genes between the injured animals that had the treatment or that did not? And that's this many. And if you look at the ones that are common to both sets, the vast majority, over 90% of them, had gone in one direction in injury and returned towards uninjured conditions in the SAM treatment. So this is a heat map that demonstrates that visually, where each row is, in this case, it is a block of, it's like 150 base pairs, the average methylation, so it's kind of tiled, and it's the average in each group, but you can see clearly, just based on the visual, that if this is the pattern in uninjured animals, it is changed in injured, but it looks in the SAM treated more like the uninjured. So the body seems to know, biology seems to know more than we do perhaps about where to utilize this extra SAM to produce the therapeutic effect. In terms of interesting pathways that we've seen, cell signaling and communication, there were a lot of genes in that like reversal set related to cell signaling. There were genes related to cell motility, and a large number, quite a strong presence of biological functions related to actin cytoskeleton, which was really interesting and would have been really surprising if Dr. Diachenko and her team had not already shown that in RNA-seq experiments in chronic rodent models maybe 2 years ago. We also did a running experiment, which I'll go over quickly, in the nerve injured animals, just to show you that we also got a similar kind of pattern of reversal of the pain injury effects in the therapeutic exercise. And really interestingly, of all of the top biological pathways that were represented here, all of the ones highlighted right now are related to vascular endothelial growth factor, which is really interesting because that implies that the voluntary running is increasing vascularity and that is therapeutically beneficial. So we're definitely planning to follow up on that. Finally, in the last few minutes, I'm going to talk about some of the human work. So if we're going to use any of this information to improve outcomes, it really needs to be taken from preclinical models into clinic and into clinical practice and into clinical studies. So that's what I'm going to talk about here. And there's a lot of things that we can study clinically. We can look at some of these risk and resilience factors. Maybe they might be useful biomarkers. We can try to understand the epigenetics of acute pathology and how to block that before pain even happens. Or maybe reverse with a treatment the impact of that chronic pain on the organism. But we really need to go to humans. So I've had the great fortune over the last 10 years to work with this group from the Quebec Pain Research Network, where I co-founded and then later on directed the Quebec low back pain study. And Dr. Lutz referred to this early on in the introduction. So I'll just briefly summarize. We're using a two-tiered strategy where the first tier is an online longitudinal database where we collect biopsychosocial data based on questionnaires at regular intervals over a 2-year period. The second tier comes from the collection of biological samples, which might be used for genetic or epigenetic or microbiome work, for example, as well as satellite projects where researchers or partner groups such as Backpack might work with us to access data or participants to further other research goals. To date, we have recruited approximately 330 individuals in the acute back pain stage and are following them. And we've almost hit 4,000 individuals total, the vast majority of which had chronic pain at the time that they enrolled, but many are still enrolled. Our 12-month numbers are very encouraging at almost 1,000. So we're very excited about that. And I'm going to tell you a few things that we've learned from this project so far. So the first question, the most basic question that kind of predicates whether there's anything here to study at all, is there an epigenetic signature of chronic low back pain in humans? And in order to ask this question, we recruited men and women, either pain-free controls or with chronic back pain. And we divided them into a discovery cohort and a validation cohort. And we gave some standardized questionnaires to measure pain, anxiety, disability, collected blood, isolated T cells, and then ultimately performed epigenome-wide mapping studies. And what we found was the short answer is that yes, we do believe there is an epigenetic signature of chronic low back pain in humans. Something very interesting was observed, which was that the pattern in males is completely different to the pattern in females. And if we analyzed all the data together and didn't separate by sex, the results are very different than if we separated them. They were much less interesting together because I think that they wash each other out. Some of the major pathways or biological functions included actin, cytoskeleton, and the immune system. So there are some themes emerging here. And before we've had a chance to publish this, another group reported a similar study, which definitely deserves to be mentioned here. So Edwin Roque, I hope I pronounced that right, did an analogous study, and they found also immune signaling as one of their primary biological themes in their data set. So there's some really, there's a lot of work to do understanding how these changes in methylation might map to disability or pain intensity or comorbidities of anxiety and depression and so forth, but there does seem to be something here that we can work with. And then what I believe is the last data slide, we've also been starting to play with the idea of epigenetic biomarkers for chronic back pain or chronic pain in general. So if you remember when we did in the rat study, we identified 11 different sites in the genome whose methylation status could predict whether an animal had received a nerve injury or not. So this is what we're trying now to do in humans. We're at very early stages, so please keep that in mind. This is a proof of concept at this point. But this is a heat map showing 3 individual CPGs. Each row is an individual methylation site, and the color indicates the amount of methylation. So you can see on this side are healthy males without pain, and this side are males with pain. And these 3 CPG sites alone, if you combine them into a polygenic score mathematically, you're able to see a really big separation between the methylation status in controls and in individuals with low back pain. But interestingly, the male methylation score has no predictive power in females at all. And we see the same thing when we've made a female score. It has no predictive power in males at all. They're completely independent. My epigenetics collaborator, Dr. Moishe Sif, says that I didn't believe him at first when we were doing the experimental design. He said males and females are epigenetically different species. You cannot pull them. You have to, they're different species. And now I'm starting to think maybe he was right. And so where we're going with this is we have to increase our sample size so that we can use machine learning algorithms to develop better predictors of whether or not an individual has back pain, but then eventually whether they're likely to develop back pain. And so what we have at this point is a probability score based on just a few CPGs in males, where 0 means you're not likely to have pain and 1 means you are. And so this is an early machine learning test. We definitely need more subjects, but it's pretty encouraging because a lot of the probability predictor got a lot of the individuals right. But this is very preliminary. So that's, I think I've said that it's preliminary about 10 times because I'm very nervous about, it's gonna, I'm sure it's gonna change as we increase the sample size. So to conclude, I think all along the pain life cycle, there are opportunities either to target epigenetic mechanisms therapeutically, to study epigenetics to understand possible other ways to target pain-driving mechanisms, as well as the potential for biomarkers. So what we're doing right now, in addition to refining the predictive validity of whether we can tell if a person has or does not have back pain, is can we tell at the acute pain stage from a methylation pattern who's most likely to develop chronic pain? Or can we tell in individuals with chronic pain who's most likely to respond to one treatment or another? So we currently have a study, an acute to chronic pain study, where we're collecting samples with the Quebec back pain study, as well as a exercise intervention study in humans being run by Dr. Mathieu Roy, in back pain patients, where we're going to see if the epigenetic signature shifts towards a non-pain phenotype after therapeutic exercise in humans. And with that, there's many, many people to thank, to the point where I think I will just save the time for questions, but thank you everyone who's been involved in all of this work. I've been so lucky to have amazing collaborators and team members. Thank you very much.
Thank you so much, Laura. It was a very interesting presentation. So those of you who have questions, please enter them into the chat. I have a quick question as we get started and thinking about interactions between what you're seeing in terms of methylation patterns and treatments. And you mentioned running. And do you anticipate that there'll be these synergistic effects between the patterns you see in the patients and a particular intervention that you might match them to?
That is our 10,000 foot kind of hope, is that we'll eventually get to the point where we can understand these patterns sufficiently to recognize whether a certain individual will be best served with a psychological intervention, or with this therapeutic drug, or with yoga or meditation, or all of the, so that we can help to better match individuals to how they're most likely to respond. And it kind of makes sense, because we know, for example, that depression and anxiety have both been associated with their own methylation patterns in blood. So an individual might have a pattern which suggests that anxiety is a big driver, or might be a big driver of their pain, so that's something that should really be focused on. Or maybe it's a biological change, like something to do with collagen or aggregan, where maybe that would predict a different kind of treatment. So, I mean, that's kind of our big, it's a bit of a dream right now, but that's where we're going towards. And I think that's very much where Backpack is going towards as well, is can we use information about patients to help them get better faster, or even better yet, to prevent chronic pain in the first place. So epigenetics might just be one of the many things that go into a set of data that can help us do better.
Okay, thank you. There's a question that was posted about if you have data on histone markers or histone acetylation as well.
No, we have not looked at that at all. There is literature from preclinical models that targeting different histone modifications can be used therapeutically, but it's very early stages.
Thank you. There's another question that kind of relates to the specificity of these markers relative to the source of pain. So what's a question about studying cancer pain? Does that look different than back pain or knee pain or hip pain?
The short answer is we have no idea. The slightly more sophisticated answer is what I suspect we're going to find, where there are some common patterns amongst all chronic pain conditions, but that each condition has its own unique pattern. So the things like if we were to try to come up with a generic biomarker for all chronic pain, what we would need to do is find those common threads that represent pain as an experience, rather than just nerve injury or disc degeneration or
right.
So
it seems like this epigenetic plasticity would be a kind of a companion therapy. So if you have a nerve root injury, you want to have a discectomy to get rid of the nerve compression in combination with some kind of epigenetic approach. Is that kind of what you're thinking, that there's different targets for the interventions that would be customizable?
Customizable, yeah. And the epigenetics might also help us to understand which therapeutic approaches will be best in what combinations.
Uh-huh.
But at a minimum, so again, that's the high, the 10,000-foot view, but at a minimum, by studying epigenetics, we're going to understand more about the pathologies underlying chronic pain. And that itself might lead to insights into new approaches.
Okay. There's a question here, kind of a practical question about the type of blood samples that are used for this analysis. Is it full blood or is it fractionated? Are you looking at T cells or?
Right, so in the work that I showed you, we were isolating T cells, but we are now switching to using just peripheral blood mononucleocytes just altogether, because there's now bioinformatic methods that can be used to try to understand the contribution of different cell types. It's not as clean as if you look at different cell types, but we're moving towards it. And in the study that was published by the group in Alabama that I showed, they used just PBMCs.
Okay. And I think we have one last question here, and then we'll finish. Based on your data, would you say that propensity to feel pain with high intensity is genetic, or rather it's characteristic of epigenetic life experiences?
That is a completely loaded question. Right? So we very much just went back to the original nature versus nurture debate that's been going on for forever. And I think it's a mix, which is, I realize is an unsatisfying answer, but it's not all genetics, and it's not all environment and life experience.
Right. So we need to be able to look at all of these things in combination.
Absolutely, yeah.
Okay, thank you so much, Laura. I want to be respectful of everybody's time. This was a really interesting, thought-provoking presentation. I learned a lot, and I'm sure everybody else did as well. So we appreciate that and look forward to working together with you.
Thank you very much for your time, everybody.
Thanks, everybody.